Hello, this is Gina Piscatelli with another lecture for Anatomy and Physiology 2 at Madison Area Technical College. In this lecture, we'll look at how cardiac muscle cells contract. That's the cardiac muscle physiology part. And then we'll also look at electrocardiograms and find out exactly what an ECG measures. So first, let's start with cardiac muscle physiology. How does the muscle contract? So contraction of the myocardium is going to require depolarization first. You know this already because you know that depolarization or excitation is coupled to contraction. So depolarization is going to spread from the cells of that intrinsic conductive system in the heart, the nodes, the bundles, the bundle branches, and the Purkinje fibers. It's going to spread, the depolarization is going to spread to the actual contractile cells. This spread happens from cell to cell through gap junction. In addition, once a set of contractile cells in the myocardium have been depolarized, they can also spread the depolarization to neighboring cells. And so that depolarization is going to spread from one contractile cell to another through gap junctions again, but those are located in intercalated discs, which you've seen under the microscope when we looked at cardiac muscle histology. So this is one of the theories of the way that depolarization is spread from cell to cell within the heart, whether it be from a non-conductive cell, a non-contractile cell to a contractile cell, or between two cells that contract, whatever two cells we're talking about. It's thought that these gap junctions function as channels made up of specialized proteins called connexins. Doesn't matter. The ions then can move from one cell to another. And if it's sodium, for example, the inside of the cell will become more positive and be depolarized. So that's how the depolarization spreads. What does the depolarization stimulate? Well, once depolarization reaches a threshold level, it's going to stimulate an action potential in a contractile cell. Now this is the action potential that I'm going to outline. It's in dark blue. So at rest, and then this is the beginning of the action potential, and then it comes down. So I've just traced it in green. Okay, that's an action potential. And the action potential will function to stimulate the release of calcium from the sarcoplasmic reticulum of the cell. And that's going to stimulate the contraction. So the contractive force is shown in this graph with the line in purple. So the force builds as the purple line ascends, it reaches a peak, and then it goes down. Notice that there is a delay between depolarization and contraction, or from the action potential to contraction. The action potential always has to happen first, and then you get contraction, which is second. If anything blocks the depolarization, you will not get contraction. Okay, so continuing on with the electrophysiology of muscle cells in the heart, we're going to look at what's happening in the action potential of in a contractile cardiocyte. So this is a weird looking action potential. We've got a straight, nice resting membrane potential, and we have a very rapid depolarization, and repolarization looks pretty normal. But notice this plateau phase, number 5. Okay, that plateau phase is unique for heart muscle cells. And that doesn't reflect contraction per se. It's reflecting how long the cell remains depolarized at a maximum level. So we haven't seen that before. Let's look at the ion movement that happens in and out of the cell at each step. So at rest, this resting membrane potential, we have some potassium leaking out, but not a lot. So basically, we'll just say there's very little. There's a little bit of potassium movement, but that's the only thing that's moving. And for our purposes, it won't make any difference. But notice that the resting membrane potential is quite low, lower than in either a neuron or in a conductive heart cell, like in the SA node. So negative 90 millivolts is the lowest. Okay, then once threshold has been reached, and threshold's not actually shown on here, we get rapid influx of sodium. And sodium continues to go into the cell, but that will cause some opening of membrane calcium channels. So calcium is also going to go in. So we have both sodium and calcium influx during depolarization. Now at number 4, those sodium channels are going to close. The calcium channels stay open a little bit longer, but what's really important is this high level of depolarization will cause channels in the sarcoplasmic reticulum that are specific for calcium to open. So now calcium is going to flood the cytoplasm of the muscle cell. And so that makes the inside of the muscle cell stay positive, or the cytoplasm anyway, stay positive for a longer period of time. And you know we need that calcium for maximum numbers of cross bridges to form as well. So this just reflects that calcium is flooding the cytoplasm. And then during the repolarization event, we have the ion that moves the most is the potassium. So just like in every other cell type we've seen, potassium leaving causes the repolarization event. So it's not crucial, I should say, that you understand this graph of the separate ion movements. But I do want to show you that it does correlate to what I think you already understand. So in this depolarization phase, we have a lot of sodium movement. And here we go. That's a high peak for sodium, right? During the plateau phase, we have a lot of calcium leaving the sarcoplasmic reticulum, and that correlates to the high degree of calcium movement shown here. And then in the repolarization phase, we have a lot of potassium movement, and that's another peak shown right there. So while I don't think you'll never see this particular graph, the one on the bottom, on a test, it does correlate to what you know. Okay, so this is a summary of everything that I just mentioned to you, and I'm just going to highlight a few of the important points. So the resting membrane potential of a contractile heart cell is negative 90 millivolts. That's unique. Threshold is negative 70. I don't really know if it matters to me that you know that. Sodium and calcium channels open, and so that's what accounts for depolarization. Calcium channels stay open during the plateau phase, but more channels that are in the membrane of the sarcoplasmic reticulum open, and you get an influx of calcium into the cytoplasm. At the end of the plateau phase, all calcium channels in the membrane of the cell close, and the sarcoplasmic reticulum pumps cytoplasmic calcium back into its organelle. And then finally we have potassium leaving during repolarization. So that's the action potential or depolarizing event that is required for a cardiomyocyte to contract. Now we're going to look again at electrophysiology, but this is going to be a much bigger picture topic. So this is the second topic of the lecture, and it's analyzing the electrocardiogram or ECG. So many of you have seen these ECG tracings before, if nothing else on television. And you see these little blips, these peaks. Please, right now, I want you to listen to this for a second. Those are not action potentials. Okay, this is not an action potential. Not an action potential. Instead, what an ECG is doing is measuring all electrical currents that it picks up. So we place electrodes on the skin to pick up electrical currents that are going through all parts of the heart. You could place electrodes in different locations of the body, like on a quadricep muscle, and then have it contract, and you would get an ECG reading. But we put it around the skin of the chest, and all these electrodes pick up signals that emanate from an intersection in the middle here. So that means that all action potentials and all electrical currents are being picked up, summarized or added together, and then you get a picture of this tracing. Okay, so all electrical currents are being detected, both contractile cells and non-contractile cells, over time. And then a graph is produced, voltage over time. So let's look at the waveforms that you see. Now that I've stressed that this is not an action potential, we still do see some deflections that indicate there's more or less current occurring, right? So you see a deflection labeled P. You see a deflection labeled QRS segment, and then a T. Sometimes there's a U, sometimes there isn't. I mean, visible. There's probably always a current, but visible. So what these deflections relate to is the intensity of a current at a given point in time, of all currents at a given point in time. So we see this upward deflection indicating there's an increase in current activity, right, electrical activity, and that correlates with the SA node depolarizing the atria. So at the same time, we're going to see that the atria depolarize right here and spread that depolarization. The QRS segment is a higher peak. There's more cells involved and therefore more currents, and it correlates to when the ventricles are depolarizing, but at the same time the atria are repolarizing. So you have a lot of outward current movement, inward current movements in terms of cell position. But nonetheless, the QRS segment is when the ventricles depolarize and the atria repolarize. Now in the T wave, that's the time period when potassium is leaving the ventricular cell, but it's happening in a lot of cells, and therefore ventricles are repolarizing during this T wave. The U wave has to do with the papillary muscle, which is attached to a chordae tendineae of a valve. And we didn't talk about when that depolarizes, but it's going to be somewhere in the QRS segment. I'm not sure exactly early or late. Okay, so in the P wave, this is another graphical representation. In the P wave, we have the SA node starting the depolarization, and then in the interval between the peak of the P wave and the QRS segment, all ventricular cells have all atrial cells have depolarized. Then we reach the QRS segment, and you'll notice that during the QRS segment or complex, the interventricular septum and the apex of the ventricle is beginning to depolarize, and then eventually towards the end of S, all of the ventricle is depolarized. Now it's going to take some time for repolarization to happen, but repolarization begins at the apex as well and moves up superiorly, and that correlates to the T wave. Now in lab, we're going to get a chance to work a little bit with ECGs and calculate heart rates using ECG, so I just want to introduce it to you. This tracing has no graph on it, but if it did, and if it was real, it would have lasted 6 seconds. Okay, so one large box is 0.2 seconds, not that that matters. And when we calculate heart rate, we do it per minute, right? Which is 60 seconds. So if I were to draw a number of, okay, there's a block there, but a number of waves, we'll just call them waves, and let's say there were 4 here. You know that you got 4 QRSs in 6 seconds. So how could you, one QRS is 1 heartbeat, right? Because that's when the ventricle depolarizes and then contracts. So you could figure out the heart rate just multiplying by 10 for the time. 6 times 10 is 60 seconds, and the number of times the heart pumped would be 40, which is a very low heart rate, but that's because I can only draw so fast. So let's look at a real one. Okay, so here's an example of a heart rate, an ECG tracing over 6 seconds, and we usually count the R points here. So there's 7 of those in 6 seconds. So how many would there be in 60 seconds? We just multiply by 10, and so you'd get 70 beats per minute. Now, just so you know, this is electrical right here, this graph, but we're using the electrical to predict the number of times the heart physically contracts, and you would see movement. Okay. Now, I've got one quick question for you here. If this ECG was taken while someone was exercising, how do you think the R to R interval would change? And we will answer that in lab. Okay, let's look at some abnormal ECG tracings. So this is a normal sinus rhythm up on top. And down below is something called a junctional rhythm or a nodal rhythm. Nodal refers to the AV node, not the SA node. So maybe junctional is better. Anyway, this occurs when the SA node is not functional. And you can tell this because we've got a nice QRS. Here's R up here. And after the QRS segment, we do have our T wave. But where's the P wave? There is no P wave. So that means that the SA node is not sending a signal to the atria. It's not functional. So the AV node is going to set the rate. And here is where you see the example that I showed you before of 40 to 60 beats per minute. That's what the AV node would set the heart rate at. Here's another example of an abnormal ECG tracing. In this case, there are more P waves than QRS waves. So I'm going to start right here, and I see a nice P, QRS, T, then a P. Oh, whoops, where's the QRS? There's no QRS, right? So there's some kind of block to the ventricles. So the AV node is, or something in the bundle, the AV bundle, is failing to conduct some impulses to the ventricles. So this is what we call a second degree block. Not that you have to memorize that, but okay. Here's what fibrillation looks like. It's just electrical activity completely disorganized. Action potentials occur randomly in different locations in the heart. There's not a nice sequence of events anatomically, and so the electrical currents are kind of all over the place. And this is seen during a heart attack or after an electrical shock. In the ventricle, pretty severely. In the atrium, also severely, but this shows me that it's possible that the AV node is still working. Okay. So that's all for the ECG lecture and the cardiac muscle physiology. Thank you for listening.